Good morning, students. Welcome back to biology class, chapter 10, cell cycle and cell division. You know that all organisms start their life from a single cell. Okay, you know the last class you have studied what is a cell. Cell is the structural and functional unit of the living organisms. Growth and reproduction are the characteristics of cells, indeed of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. Cell division is a very important process in all living organisms. During the cell division of a cell, DNA replication and cell growth also takes place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two daughter cells is termed cell cycle. Although cell growth in terms of cytoplasmic increase is a continuation process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosome DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. What are the phases of cell cycle? A typical eukaryotic cell cycle is illustrated by human cells in culture. These cells divide once in approximately every 24 hours. This duration of cell cycle can vary from organisms to organism and also from cell type to cell type. Yeast and its cell cycle is only about 90 minutes. Human being cells 24 hours and in the yeast cell 90 minutes. The cell cycle is divided into two basic phases. First one interphase, second one M phase, that means mitosis phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phases. It is significant to note that in the 24-hour average duration of cell cycle of a human cell, cell division proper lasts for only about half an hour time. The interphase lasts more than 95% of the duration of cell cycle. The M phase starts with the nuclear division corresponding to the separation of daughter chromosomes, karyokinesis, and usually ends with the division of cytoplasm, cytokinesis. Okay. Division of cytoplasm is called cytokinesis. Division of nucleus is called karyokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases. First one, G1 phase. The G means a gap 1. Second, S phase, that is the synthesis phase. Then the third point, G2 phase, gap 2. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. See, listen, G1 phase corresponds to the interval between mitosis and the initiation of the DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. S or synthesis phase marks the period during which DNA synthesis or replication takes place. Okay, replication takes place in the synthesis phase, S phase. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted to 2C, then it increases to 4C, doubling. However, there is no increase in the chromosome number if the cell had diploid or 2n number of chromosomes at G1 phase. Even after S phase, the number of chromosomes remains the same, that is the 2n. In animal cells, during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Some cells in the adult animals do not appear to exhibit division. Example, heart cells. And many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells that do not divide further exit G1 phase. To enter an inactive stage is called quiescent stage or G0. Quiescent stage, okay, of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so depending on the requirement of the organism. In animals, mitotic cell division is only seen in the diploid somatic cells, body cells. Against this, the plants can show mitotic division in both haploid and diploid cells. From your recollection of examples of alternation of generation in plants, plant species can identify and stages at which mitosis is seen in haploid cells.